Welcome, okay, yeah, we are live. Welcome everyone to Flora Fauna Science Foundation's Wave Talk series, part of our Science to Smile series. Today we are honored to have Dr. Ashutosh Shukla, Senior Principal Scientist from CSIR CMAP, who will share insights on the fascinating journey of Periwinkle, from different traditional knowledge to modern medicine. We are also privileged to be joined by Professor S.P.S. Khanuja, Founder Chairman of Flora Fauna Science Foundation, and Dr. A.K. Singh, Vice President of the body. I also welcome our team members who are too in the forum today. So without further ado, let us begin the session with opening remarks of Professor Khanuja. So please.
Thank you, Dr. Manika. A very warm welcome to all those who are listening and viewing this lead talk by Dr. Ashutosh Shukla. This platform of Flora Fauna Science Foundation, which holds the talks by experts in various fields according to the theme areas of Flora Fauna Science Foundation, uh, is always on for disseminating the knowledge, or we can say translating the science for society. And our main focus always remains villages. Uh, I must, in the beginning itself, thank Dr. Ashutosh Shukla for having agreed to deliver this very important talk. I'm also privileged to say that Ashutosh is my student who did PhD with me, and therefore my collinearity with his area of knowledge is almost plus 90%. आप सभी का फ्लोरा फोना साइंस फाउंडेशन के इस मंच पर स्वागत है। आज का ये जो व्याख्यान है डॉक्टर आशुतोष शुक्ला जो सीमैप में कार्यरत हैं उनके द्वारा दिया जाएगा। और जैसा कि आप इसके विषय में देख सकते हैं, ये सदाबहार पर है, जिसे पेरिविंकल या कैथेरेंथस के नाम से भी जाना जाता है। तो आज हम विशेष तौर पर किस तरीके से विज्ञान का जो ज्ञान है वो समाज के लिए लाभकारी हो सकता है और स्वास्थ्य के लिए, यानी कि ड्रग वैल्यू या औषधीय जो इसके गुण हैं उसको आप तक पहुंचाने की कोशिश करेंगे। इससे पहले कि हम विषय विशेष पर जाएं, मैं आपको बताना चाहूँगा कि फ्लोरा फोना साइंस फाउंडेशन एक मिशन पर काम करती है, और उस मिशन को हम ई विलेजेस बोलते हैं, एनेबल्ड एंटरप्रेन्योरियल विलेजेस, जिसमें हम ये चाहते हैं कि विज्ञान द्वारा जो खोजें होती हैं वो गांव-गांव तक पहुँचे और गांव में उसके अपनाने से किस तरीके से खुशहाली आ सके, और उस खुशहाली का जो कारण है वो ना केवल आर्थिकी हो, उसके साथ साथ पोषण और जो स्वास्थ्य है वो भी लाभ उठा सके। तो इस तरीके से हम हर वर्ष एक थीम या एक विषय लेकर चलते हैं। इस वर्ष का जो विषय है या थीम है वो है विज्ञान से मुस्कान। द थीम ऑफ दिस ईयर इज related to how science can spread smiles in society. So smiles from science, or we can say science to smiles. And therefore, the reason for those smiles has to be knowledge that gets translated and converted into technologies that everyone can use. Today's topic is going to be useful for researchers, for students, for society, and also the farmers and villages when it just gets translated. We deliberately chose Dr. Ashutosh Shukla for this topic because he is a man who has almost spent the academic life on this plant, and therefore would be able to translate the findings of this and linking those findings with what traditional knowledge says. जैसा कि आप जानते हैं, पेरिविंकल या कैथेरेंथस को सदाबहार के नाम से जानते हैं। उसका एक कारण है, इसका कारण है कि ये हमेशा बहार में रहता है। एक तरीका सोचने का बहार का है कि जिस पौधे पर जब फूल आ जाते हैं तो हम ये कहते हैं कि उसमें बहार आ गई, और वो फूल आपको विशेष तौर पर सफ़ेद और गुलाबी, पिंक एंड वाइट, आप देखते होंगे ना उगाने से भी ये उग जाता है। इसलिए इसकी जो सदाबहारता है वो केवल खेतों में नहीं, बल्कि खेतों ने नेचर से या प्रकृति से सीखा कि ये कहीं भी अपने आप उग जाता है और इसकी कोई विशेष मांग भी नहीं होती। My way of thinking that this is Sadabahar, that means evergreen or ever flowering or ever fruiting for the benefit of the society. It must translate into it in the sense that it's it's being ever usefulness. Not only is in modern medicine where we talk about its uses, particularly in cancer therapies, but also in almost all cardiovascular or almost all lifestyle diseases which are now becoming too prevalent in the society. So this lifestyle diseases, whether they are cardiovascular, whether they are fat and sugar metabolism, whether they are related to brain or they are related to hormonal controls, this plant finds uses. Not only that, this plant also has a beauty that all its parts are useful. So this is another reason to call it Sadabahar, that there is no part of it which is not useful. The only thing that we have to learn is when is which part useful, and that is what the main work of Dr. Ashutosh is, where he has been able to do at molecular level through molecular biology and genetics approaches, that where are the genes expressed for which metabolism, एंड देन दैट दोज मेटाबोलाइट्स जिनको हम ड्रग्स या औषधीय कहते हैं, औषधीय गुण कहते हैं, वो वहाँ पर एकत्रित होते हैं. द phytoceuticals or phytopharmaceuticals get accumulated in various parts at various stages, and that how do we know is the main work that Ashutosh has done. We have been working on theme areas, and today's theme, as I told you, is Vigyan Se Muskan. Uh, Before this, we had Fasal Se Safal like theme, and then Krishi Se Kushi, Poshan Se Roshan, Khet Se Sehat. So always you will see that we take one prefix and then say from that prefix to what is the suffix. That means the translation of the science into knowledge and then ultimately entrepreneurship. And for this, we also have a magazine or the messenger of Flora Fauna Science Foundation called Flora Sarthi, where our members as well as people from outside society and other organizations also contribute articles which are useful to everyone. So you do look at this at it is available in digital form on our website and also on requesting to Dr. A.K. Singh or our Flora Fauna Science Foundation itself. Dr. A.K. Singh happens to be also the chief editor of Flora Fauna Science Foundation, in addition to being the vice president of our foundation. And to do this, to translate this, we also have taken up certain activities like we have gone for Nutri Vatika, where we look for the plants that are useful for society, which give nutrition. पोषण के जो पौधे हैं उनको एक साथ उगाकर उनको इस तरीके से लगाना कि हम समझ सकें उनके क्या गुण हैं जिससे स्वास्थ्य लाभ हो सकता है. और इसीलिए हम ये कहते हैं कि जो हमारा विजन है इस मिशन को अचीव करने के लिए वो है ब्रिंगिंग health and through health and nutrition using science, we want to make a society which is enabled society. So that vision and this mission is getting translated by the expert talks that you keep on hearing. And today we will have Dr. Ashutosh, who would simplify for all of us the use of periwinkle, जो सदाबहार के उपयोग हैं उनको कैसे हम एक दूसरे से संबंध भी बता सकते हैं कि जो हमारा पारंपरिक ज्ञान है और जो आज के जो आधुनिक गुण हैं, औषधीय गुण हैं, उनसे किस तरीके से आपस में संबंध बनाकर हम इसका उपयोग कर सकते हैं. मैं और अधिक समय ना लेते हुए डॉक्टर मनिका से कहूँगा कि वो डॉक्टर आशुतोष शुक्ला का एक औपचारिक परिचय भी दें. ए फॉर्मल इंट्रोडक्शन ऑफ डॉक्टर आशुतोष शुक्ला. Thank you.
Thank you, sir. It's a privilege to introduce Dr. Shukla as he's my mentor too. Dr. Shukla is a senior principal scientist in the Biotechnology Division of CSIR CMAP, Central Institute of Medicinal and Aromatic Plants, Lucknow. His primary education started in Washington, D.C., USA, and he got his higher education in Lucknow, India. He holds a PhD in biochemistry and MBA marketing from Lucknow University. He specializes in medicinal plant molecular biology with a focus on alkaloid producing plants. His work has provided new insights for omics based characterization of secondary metabolism in contrasting organs, genotypes, and development stages of medicinal plants like Catharanthus roseus and Pepperus somniferum. He has widely traveled abroad and has worked in various laboratories as visiting scientist. He has been an IUSSTF Research Fellow at the Donald Danforth Plant Science Research Center, St. Louis, USA, and a Raman Research Fellow at the Albert Louis University, Freiburg, Germany. He has received many prestigious awards and fellowships in his career, prominent among which are Professor P.S. Krishnan Memorial Gold Medal for securing first position in order of merit in the MSc Biochemistry examination conducted by University of Lucknow in 1996, Biotalent Award twice at Bangalore Bio in 2007 and 2008, the Indo-US Science and Technology Forum Research Fellowship 2012, and the Raman Research Fellowship for 2017-18 from CSIR. He has 77 publications, contributed to 9 patents, 3 technologies, and 4 plant varieties, as well as edited 2 laboratory manuals. He is also life member of many prestigious societies. He has convened 4 school trainings and have been involved in 34 projects. Is reviewer for many international journals and is on editorial board of Journal of Medicine and Aromatic Plant Sciences. Since 2022, he is serving as member Program Advisory Committee PAC SERB DST for a special call on biology of plants under extreme environments and co-opt member of the Program Advisory Committee on Organismal and Evolutionary Biology of SERB DST. He has guided or co-guided 13 PhD scholars and mentored 17 graduate trainees. With this very brief bio, I request Dr. Ashutosh Shukla for his talk.
So thank you, Dr. Manika, very much for the nice introduction. I would like to take this, I hope I'm audible properly.
Of course, sir.
So I would like to take this opportunity, first of all, to thank the Flora Fauna Science Foundation and the founding chairman, Professor S.P.S. Kanuja, who is also my guru. So he is the one who introduced me to Sadabahar during my doctoral study days. And he has been a great inspiration for me. And I'm fortunate to have him as my teacher and his support throughout my research. My today's talk is based on this very important medicinal plant Sadabahar, as already told by Anja sir. And I will not take much time and come straight for the talk so that I finish timely also. So I hope my slides are visible. Is my screen visible, Manika?
No, sir, you have to share it again because.
Okay, I will do it.
And yes, sir, now we can see your slide. You can maximize it.
Yeah. So the
We are
talk
good to
I
go.
will be speaking is Periwinkle, the drug value of Sadabahar from traditional knowledge to modern medicine. So this plant is already described in the traditional medicine systems, but it gained prominence once it also made way into modern medicine. So in the next slide, as you would see, it comes in a wide variety. You can see catranthus or periwinkle flowers in different colors, which are range from purple to white to a combination of all these colors, and also with very variegated petals in the flowers. The chromosome number is 16. I would also like to briefly tell about the plant and its nomenclature, how it went about in history. So Catranthus belongs to the family Echocynaceae. It is popularly known as Madagascar periwinkle because it is believed to be indigenous to the island Madagascar, which is on the southern near the southern tip of Africa. It is believed to have been introduced into India by Portuguese missionaries in the middle of the 18th century via Goa. So earlier it was not present in the Indian subcontinent, but it gained entry in the 18th century only. So currently, it has acquired a pan-tropical distribution and become naturalized throughout the tropical and subtropical parts of the world. Initially, there were some taxonomic ambiguities associated with this plant because it was also known as Amocallis rosea and Lochnera rosea in literature, and most commonly Vinca rosea. The genus Vinca was established by Linnaeus in 1753 in his species Plantarum, where he distinguished the two species, Vinca major and Vinca minor. And then he later on added a third one, which was Vinca rosea as a tropical species. Now, this Vinca rosea came to be later known as Catharanthus roseus. In 1835, George Don assigned the name Catharanthus to the genus typified by Vinca rosea in his General System of Gardening and Botany. The name is derived from the Greek words katharos plus anthos. Katharos means pure and anthos means flower. So it is believed to be a pure flower due to the neatness and beauty and elegance of the floral structure. Vernacular names popular for Catharanthus in various countries. There are various names, and in the same country, also in India, different regions use different names for it. But Sadabahar, I will be using the word Sadabahar because it is a topic of the title also in the today's talk, and it also means ever blooming blossom and evergreen. Evergreen means a thing which never dies or it can grow in adverse conditions also. As sir told, there are different meanings to Sadabahar also, meaning that the plant has no part which is not useful. So that is another sense of Sadabahar. It has been used in both the Ayurvedic and traditional Chinese medicine systems for mainly the diseases like diabetes, hypertension, and leukemia. So if one visits the Kew Botanical Gardens, there it is mentioned, there is a warning also that all parts of the Madagascar periwinkle are toxic if eaten, with the medicinal compounds needing to be isolated in safe doses before being used in treatments. So this is a sort of warning to those people who like to use medicinal plants per se, but they don't know how to use them because everything is dose dependent in medicine. You cannot have anything useful. Useful thing becomes useless also if it is taken in the wrong dose. So dose is very, very important. Along with this medicinal importance of the plant, I would also like to mention here that it is a unique host for specialized microbes known as phytoplasmas. So when you store microbes in a repository, it is very difficult to store these type of microbes which are non-culturable or phytoplasmas. So Catharanthus has proven to be a very good source or as a host for these phytoplasmas to study them for any research. So coming to the traditional value of Sadabahar in Ayurveda, this slide is prepared with the help of my friend and colleague Dr. D.N. Mani, who told me that it has been mentioned in Dravyagun Vigyan. In Ayurveda, every drug is described under various heads like Gun, Ras, Veer, and Vibhag, and the Prabhav of the drug. So this Sadabahar is having a ras which is thick. Vibhag is katu. That means the taste is like very uh, not very sweet, but it is very sour. So not sour also, but what to say, uh, something like which is not tasty. Veer is Ushna. So all the drugs in Ayurveda, they are described into two categories. One is cold and one is hot. So the Ushna means it is a hot drug, and the Prabhav is given for Rak Arbut Nashan. So Rak Arbut is the term in Ayurveda which is used for describing leukemia because cancer is a disease which has been named in the recent past. But the disease existed before the modern medicine came into picture also. So traditional medicine systems like the Chinese system and Ayurveda, they have described the diseases with different names, but the symptoms are same. So in Ayurveda, the cancer has been described by the words like Arbut, Granthi, and all these words mean cancer. So Rakt Arbud stands for leukemia or blood cancer. If one reads Acharya P.V. Sharma's 2005 book, then he will find the mention of Sadabahar for this particular disease treatment, as well as mentioning that it treats the Kapha and Vata doshas. Because in Ayurveda, a healthy body is described as a balance between all the doshas, and any imbalance between the doshas create a disease status for the body. So in Ayurveda, Catranthus or Sadabahar has been described for balancing the Kapha and Vata doshas. The plant is also mentioned for treatment of diabetes, but unlike its anti-cancer activity, the anti-diabetic property is yet to be thoroughly investigated. So the traditional benefits of Sadabar, I have tried to list them out here from literature, making them simple for the not person who is not expert in that field, but he wants to know about the plant. So I have simplified everything. Anti-cancer properties, it is famous for the alkaloids, two alkaloids, vincristine and vinblastine, which have potent anti-cancer properties. Hypotensive or blood pressure lowering properties are there in the plant. Some alkaloids like agmalicine have potential to lower the blood pressure. Mainly, I will also come in the later slides, the location of these alkaloids are different in the plant. So these are mainly the root alkaloids, the anti-cancer alkaloids, and mainly the leaf alkaloids. Anti-diabetic activity is there in the plant. Plant extracts have shown potential to reduce the blood sugar levels. Antimicrobial properties, anti-inflammatory is very important property in the plant. Antioxidant effects are present in many plants, and same way in this plant also. But very important property is the wound healing property for this plant. The plant extracts have been traditionally used topically for wound healing due to their potential to promote tissue repair and reduction in inflammation. And another thing is, because of it is an anti-inflammatory activity, it is also used in traditional medicine as an astringent for treatment of bleeding disorders, mainly in nosebleeds. In the for the women patients, it also regulates menstrual flow, helps in regulating the periods, treating postnatal ailments, and subdues the pain during dysmenorrhea, and encourages easy flow during the menstrual cycle. There are other miscellaneous themes associated with its therapeutic activity, like cognitive functioning, augments skin health, and also cures respiratory anomalies. In countries like Cuba and Jamaica, it has been used, the flower extract has been used as an eye wash for the infants. Now, this is a very old treatise on the plant. This is one of the earliest one which came into picture, the Catranthus alkaloids. Anyone interested on this plant, I would like or suggest him to go through this book. It is 1975 book, and it describes the chance discovery or the serendipity which happened for the discovery of the therapeutic activity of this plant. Earlier, this plant in the 1950s, it was famous because researchers were focusing their attention. They wanted to know about the anti-diabetic effects of this plant because they learned about a tea that Jamaicans were using, and it was lowering the blood sugar levels. So two independent groups started working on this plant. One was at the University of Western Ontario, the Nobel and Beer Group, and the second one was company, Eli and Company in Indianapolis, which was Svoboda's group in USA. But neither of them could substantiate the hypoglycemic activity of the plant. So strange enough, they started to find the cause for the anti-diabetic activity, but they ended up with something else. They could not substantiate the hypoglycemic effect. But in 1958, Nobel et al. Observed a toxic reduction in the white blood cells and bone marrow depletion in rats by some fraction of the plant extracts, and eventually that led to the isolation of vinblastine. And a few years later, in 1961, Svoboda's group isolated vincristine from the plant. So these are the scientists. I would like to mention them here because they are the ones who made this plant famous. There were characters in the plant earlier, but they were brought forth by scientists. So that way, this is a encouragement for the younger generation also, that if you work hard and try to find out the cause and effect relationships, you might end up in finding something new or something which you might not even expect at all also. So Dr. Robert Nobel and Dr. Charles Baer, they were working at the University of Western Ontario, and they were credited with the discovery of vinblastine. And Dr. Gordon Svoboda, he was in the private sector. He was working for a company, Eli, and due to the sheer force of his personality, he also convinced his bosses in the company, and to market vinblastine and vincristine. So this was one of the first and earliest examples of an academia and industry handshake. Now, coming to the main anti-cancer alkaloids which are present in the plant and which are used clinically, I would like to mention three here. I would not like to confuse the audience because many researches are going on cancer, but the things which reach the market successfully and are under clinical use, I would like to highlight or focus more on them. So vinblastine, the trade name is Wellman, mainly used against Hodgkin's disease, which is a very deadly cancer. The dose limiting toxicity is leukopenia, reduction in white blood cells, and bone marrow depression. Vincristine is sold under the trade name of Oncovin. It is mainly useful against acute lymphocytic leukemia, mainly in the pediatric patients. And the dose limiting toxicity is neurotoxicity. Vinorelbine is a semi-synthetic. The earlier two were the natural products which were derived from the plant, but the third one is semi-synthetic, invented by Pierre Cotier in CNRS, France during the 1980s. The trade name is Navalbine, mainly used against small cell lung cancer, and the chemotherapy induces peripheral neuropathy. However, as a caution, one has to note that careful titration of the dose is required to achieve minimal toxicity and the maximal therapeutic benefit from these alkaloids. The base indoles are administered intravenously in the body. To reduce the toxic side effects of these drugs on the innocent bystander tissues, new approaches for drug targeting have been employed in the recent past. Monoclonal antibodies that are reactive with tumor-associated antigens have been evaluated as vehicles for delivering the toxic base indole alkaloids to tissues that are present these antigens. The therapeutic index of vincristine can also be increased significantly through the liposomal delivery systems. So like we all have mobile numbers. So when we make a call, we want to call only the person whom we want to call and not the other persons who are there whose numbers are there in our mobile. So that way targeting of the drugs is very important. The drug should reach only those cells for which it is intended, like the cancer cells. It must not disturb the cells which are normal. Otherwise, it will not lead to any benefit. It will in fact have more toxicity. So these things are important whenever you find a new drug. The toxicity is a very important part of it. So the therapeutic effect of catranthus or Sadabahar has also been celebrated by various governments. I have given an example of a French stamp that was issued in 2000 here. And I would like to mention here that vincristine and vinblastine are very similar in their structure. If you just make an overlap of them, there is only one difference, which you can see here. I have marked in the red in this right hand side figure. So there is a single substitution on the vindoline nucleus where they possess formyl and methyl groups respectively. So there is a formyl group in vincristine and the vinblastine has a methyl group there. This minor difference does not fundamentally alter the mechanism of action and tubulin binding properties of these agents, but the anti-tumor and toxicologic profiles changes significantly. So vincristine is more common for treating pediatric malignancies, which likely reflects the combination of the higher level of sensitivity of pediatric malignancies to it and to the better tolerance of high vincristine doses in children. Vinorelbine, as I told earlier, is semi-synthetic, but it has substantially greater lipophilicity, making it a better drug candidate. Although these alkaloids produce a wide range of biochemical effects in cells, they mainly interact with tubulin. So what does to do? They do a metaphase arrest during cell division. In normal mitosis, what happens in the metaphase, the chromosome, there is a mitotic spindle formation because tubulin has to polymerize, and then the story goes to the anaphase. But when the vinca alkaloids are present, these tubulin molecules fail to polymerize because of their presence. In fact, the vinca alkaloids bind to beta tubulin and inhibits its polymerization with alpha tubulin to form the microtubules. So cell division is arrested. Anaphase is never reached. So there are various curative and palliative regimens containing the catranthus alkaloids which have been mentioned. Like most of them, you will see that either vincristine or vinblastine or any analog will be there present in all of them mostly. So these are the chemotherapy regimens which are used for cancer patients for treatment. Now coming back to the plant, I would like to say that these are not the only two alkaloids which are present in the plant. There are over 130 terpenoid indole alkaloids in the plant. So it acts like a reservoir. It is known for the anti-cancer properties of these two, but and these two are costly also. You can see that the cost is around 20,000 per gram for wind blasting, and wind crystal is even costlier. These bis-indole alkaloids, why do you call them bis-indoles? Because there are two monomeric structures which come together to form these alkaloids. They are called dimeric alkaloids or bis-indole alkaloids because of the two monomers. They are also derived from indole or tryptophan. So there are two indole moieties in their structure. These two indole alkaloids, which are the monomers, they come together, but they are kept specially apart in the plant. The reason being, if they get to couple, they are highly cytotoxic. So it is difficult for the plant to also sequester and sustain. So this is also giving a clue why these dimeric alkaloids or the bis-indole alkaloids, which we use as drugs, they are present in very low amount in the plant, maybe 0.0003% on dry wet basis. So you can just imagine the amount of dry leaves which will be required to isolate a certain amount of these drugs for therapeutic effects. So it is highly improbable that the in-planta biosynthetic potential of the plant for the bis-indoles could be raised beyond this critical threshold, because then they become cytotoxic for the plant also. And the plant is difficult, it becomes difficult for the plant to survive itself. If you see on the right panel on this slide, I have shown how these alkaloids are formed. Very briefly, I have tried to summarize. This is a very long pathway over around 40 steps. So the pathway for the biosynthesis of these alkaloids starts from two directions. One is tryptophan, which is an essential amino acid, an aromatic amino acid having an indole moiety. And the other one comes from the terpenoid moiety, which is geraniol, giving rise to cyclognin. Now, cyclognin and tryptamine, they couple to form a very important central molecule known as strictocidine. So this strictocidine gives rise to over 3,000 different terpenoid indole alkaloids in different tissues of the plant as well as other species which produce these alkaloids. So it is a central molecule. This again raises a very important research question. Too many products from a single molecule. How does it happen? So this should be a course for future research by researchers who want to know the chameleon-like nature of strictocidine. Again, strictocidine goes into different directions in Catranthus. In the root alkaloids, you will see agmelicine and serpentine are formed. And in the shoot, I have shown vindoline is the main one. And again, vindoline couples with catharanthine, which is produced throughout the plant and gives rise to windblastine and windcrystal. So you can see strictocidine I mentioned as a chemical chameleon because it is a precursor for many species specific alkaloids in different species also. Like in Rauwolfia serpentina, sarpgantha, which is like a sister or sibling of Catharanthus belonging to the same family and producing similar type of alkaloids, but with varying therapeutic activities. Similarly, Strychnos nux vomica is another plant which produces terpenoid indole alkaloids, strychnine used as a rat poison. And so there are different species producing different TIAs, which I call in short terpenoid indole alkaloids, from the central molecule, which is strictocidine. So in Catharanthus, as I told, the different tissues produce different profile of TIS. This is a very old slide which I made with under the guidance of Dr. Khanuja. He told me to classify the alkaloids based on this tissue specificity. Some of the alkaloids you would see they are specific for the shoot tissue. They are produced specifically in the leaf, not in other parts. The others are specific for the root. They are not produced in the shoot part. Some are produced in both the tissues, and some of the alkaloids are produced in tissues like seeds and other specific tissues like flowers or very specific parts of the plant, which I have mentioned at the bottom. So you can see in this slide, catharanthine, one of the monomers which forms the important bis-indole alkaloids, is produced in both the tissues, root as well as leaf. But vindoline is leaf specific or shoot specific. Why, I will tell you in the later slide, but that makes windcrystal and windblastine also leaf specific because they need vindoline for their structure. Now, age matters very much. If you go by the age of the plant, I studied the plant at 6 days old, 6 weeks age, and also 6 months old. You will see the younger plant is having higher alkaloid producing potential, both in the plumule, in the radicle, and in the mature plant in the root and shoot systems. So we try to work out the chronomics of the biosynthesis of these alkaloids under various seasons, under various ontogenic conditions, different leaves at different levels of the plant. So we came out with very interesting findings. The chronomics, as I told, as the plant grows older, the biosynthetic capability like alkaloid accumulation on dry wet basis goes down. In the seasons, if you see, the bisindole alkaloids are produced maximally under North Indian conditions. We checked in Lucknow conditions, it goes maximally in the months of June and summer. We could find the bisindole alkaloids, windbreaching and windblasting in the plant. But vindoline was accumulating at a higher level in the winter months, winter and spring. Catlanthine was somewhat phased out equally throughout the year. If you go to the ontogenic levels of the leaves, then you will find that the younger leaves are producing more of the monomers like vindoline and catlanthine, but the mature leaves are having more of the bisindoles. So this again raised a question which we tried to answer in the later researches. I will be coming to that, why the younger ones produce more of the monomers and why the mature ones need more of the dimers. Again, the form also matters, like in the case of seeing in the human body also, the form of the plant cells also matter. Differentiation is a key step for the production of these type of alkaloids in the plant. If you move from a suspension culture to a callus, compact callus, and then to a plantlet, you would see that vindoline biosynthetic potential increased as cellular differentiation increased. Now, this is a very important paper which I would like to mention here because it is an eye opener. The research on this plant was going in a wrong direction for around 35 to 40 years, and people were trying to work different strategies to produce the bisindole alkaloids in the plant because they were interested in the drug compounds. They wanted to use them as drugs and have the plant produce more for them. But the plant is not producing it for us. The plant is producing these type of molecules for itself under various stress conditions. So this group of Dr. Ropke, actually he was working in Professor D. Lucas group, who is a Canadian researcher who specializes on this plant and has done pioneering researches. They could find out in a PNAS paper that these two monomers are kept spatially apart in the plant. So catranthine, as you would see, moves into the cell surface in the leaf wax exudates, whereas vindoline, the other monomer, it moves into the idioblast and lenticular cells and gets stored in the vacuole. So the plant keeps these monomers apart and they get to couple only when there is a need. So that need arises only under a condition of herbivory or stress when there is tissue disruption and the monomers merge together or converge to form the bisindoles. So that is the reason why the dimers are not formed in a higher amount in the plant because they are cytotoxic. They are needed only at a certain point of time under stress conditions or predatory conditions by a herbivore. So as and when required by the plant, it will produce the bisindole alkaloids, not when we require it. So we have to understand these things that humans should not be selfish also. Again, I told about the monomers, that earlier paper by the D. Lucas group that highlighted the monomers more than the dimers. So the research shifted from the dimers to the monomers. So our group at that time also started working on vindoline because catranthine was not a problem at all. It was not the bottleneck compound. Production of vindoline was mainly the main bottleneck which we had to address as a scientist. So I'd also like to tell that there are other approaches to produce these alkaloids in heterologous systems also, but they also found that vindoline was the main challenge. So why is vindoline important? Again, because it is the more complex half of the bisindole alkaloids. It is the one which binds to the tubulin heterodimers. It is species specific. It is a key component of the bisindole alkaloids and also possesses some hypoglycemic activity of its own also. So this is the biosynthetic pathway again in a bit elaborated form where I have circled vindoline in red color. You can see it couples with catranthine to form the intermediate, which is the iminium intermediate, and then it gives rise to windblastin and finally to windcrystal. In the plant, the monomers are kept spatially apart, as I told earlier, and they get to couple only in the event of a herbivory or tissue damage. Although chemical synthesis of the bisindoles and their precursors has been reported, it is economically unviable. So the question arises, why should we grow the plant at all? If we know the structure of the compounds, we can produce them in the lab, then there is no need of the plant. But it is not so. Again, if we try to think selfishly, then it's always a problem. The plant retains its importance. Why? You can understand here. there is only one option, that is a semi-synthetic approach for the production of the base indole. You have to get catranthine and windolene. Catranthine you may produce anywhere in cell suspensions, but windolene is the major bottleneck, as I told, because it requires a green, well-differentiated tissues from the plant. So this is the typical herbivory bomb which I told. The monomers are kept specially apart, and then they get to couple only when some grazing animal tries to destroy the plant, and then to repel it, the plant produces instantaneously the basindole alkaloids. So that is a use for the plant, ecological, but humans make another use of those type of alkaloids for their therapeutic value. Now, this is another system where scientists have tried to use synthetic biology approaches to produce these alkaloids in yeast or bacterial backgrounds, not relying on the plant, but they have still a long way to go. They have got a very limited success. The 7-gene pathway from flavorsonine to windolene has been assembled in yeast, but there are bottlenecks at certain enzymes like flavorsonine 16 hydroxylase, NMT, and methyltransferase D4H. The de novo microbial biosynthesis of windolene and catranthine using a highly engineered yeast has been recently reported by Dr. Jake Isling's group in Nature 2022. This was the same group which reported artemisinic acid biosynthesis in yeast, which is a precursor for antimalarial compound artemisinin in
There seems to be some technical snag from the speaker's side. Please stand by.
Yeah, you can talk to him on phone if
Yeah. I'm trying. डॉक्टर शुक्ला से बात हुई है, उनकी तरफ कुछ टेक्निकल प्रॉब्लम आ गई है, वो अभी जल्दी ही कनेक्ट हो जाएंगे
okay.
और तब तक हम इंतजार कर सकते हैं।
Yeah.
Yes.
So I am sorry, there was some disruption in the internet.
Yes.
Am I audible and the slides are?
Yeah. Yeah, you are audible, Dr. Shukla. Please go ahead. Please go ahead.
So I was talking about Jake Isling's group. They tried to produce artemisinic acid earlier, and now they have tried to produce these bisindole alkaloids. But vinplastin was not directly produced in yeast because the peroxidase, which is the final coupling enzyme for catranthine and windolene, was not expressing in yeast. Besides, the de novo windolene production was found to be more challenging as compared to catranthine. So again, windolene was the main bottleneck compound. These are the modules which they tried to synthesize and put in the yeast. You can see the blue, the yellow, and the purple, pink, and the green colors. which define different types of host yeast strains expressing the part of the pathway which has been defined in that color. So they put all these strains together and tried to produce vinblastine, but they failed because the peroxidase shown down here was not expressing the elements. Nevertheless, this study was a proof of concept study that such compounds can be produced successfully in the yeast background, and it gives rise to thoughts that synthetic biology would one day be able to produce most of these natural products in microbial systems which are like easy to grow and economical also. But the economics need to be worked out. Till the date that is being done, it is very difficult for to bypass the plant. So the plant retains its importance, as I told earlier. Since Windolin is a major bottleneck, we thought that there is a need for identifying its genotypes that accumulate higher than usual levels of Windolin and the factors that promote its biosynthesis and accumulation. Any improvement on this front will drastically reduce the cost of the bisindole alkaloids very realistic. So in an earlier study where we had gone for a comparison of the different tissues, the leaf minus root suppression subtractive hybridization yielded certain genes which were specifically expressing in the leaf, which is also the part which expresses the alkaloids, the bisindole alkaloids and windolene. And the root alkaloids are different. They are having different activities also, and their pathway is also differing. So we try to have a leaf minus root subtraction, identify the genes and try to functionally validate some of those. The comparison of the root and leaf transcriptomes also showed that the leaf transcriptome was having distinct number of distinct transcriptionally defined fragments through cDNA FLP approach. So logic-based selection of certain candidates was done, and the basis for selection was their possible involvement in key terpenoid indole alkaloid biosynthesis, metabolism and diversion pathways and regulation. Genes like allene oxide cyclase and temperature-induced lipocalins were selected. There was also a study in parallel by our plant pathology group, which works on the plant. They found that when the plant is being a host to various phytoplasmas, it has a distinct morphology and little leaf disease sets in. Similarly, the plant is also infected by viruses. When such type of pathogens were infecting the plant started producing more amount of windolene specifically. I don't know why, but the reason was that there must be some need for this monomer to be produced in a larger amount. So that was a phenomena that was required to be studied in detail. The phytoplasma was characterized, which was infecting the plant. It belonged to 16S R2 group. Similarly, the cauliflower mosaic, the mosaic virus, which was infecting Catranthus, it is also a CMV. It was found to be belonging to subgroup 1B. So this allene oxide cyclase, I would like to very briefly tell in a very simplified manner that it co-expresses with many of the biosynthetic pathway genes which are there, which are shown in this diagram, which are ranging from tabersonin to windolene. So these genes co-express with allene oxide cyclase, which gave a hint that allene oxide cyclase, which is responsible for jasmonate biosynthesis, might also bring about a trigger for the biosynthesis of windolene in the plant, and ultimately the bisindole alkaloids. So this particular gene, allene oxide cyclase, was studied through real-time PCR, through bioinformatics approaches, and its promoter was also studied for various cis regulatory elements which are present therein. And this specific co-expression pattern was found to be there, where we could say that there was a direct or positive relationship between allene oxide cyclase, which is termed AOC in short, with windolene content in the plant, establishing this a key modulator for windolene biosynthesis. The co-expression of allene oxide cyclase with windolene pathway genes under various stresses also brought into focus another family member, temperature-induced lipocalin, because lipocalins is a very big family of proteins. It was also taken up for molecular cloning, tissue-specific expression profiling, and in silico characterization, and upstream genomic region analysis for cis-regulatory elements. Stress-mediated variations in TIL expression were reflected in the form of disturbances in cell membrane integrity assayed through measurement of electrolyte leakage and lipid peroxidation product malondialdehyde MDA, which implicated the role of CRTIL in maintenance of the cell membrane integrity. But when we tried to silence this gene in the plant and overexpress it, we found that it had an inverse relationship with the windowline content in the plant. So it raised more questions than answers, and then we had to go for a subcellular localization, and then we could validate its role and propose a model how this important lipocalin was modulating windowline content in the plant. You can see the lipocalin structure was worked out again through in silico approaches and modeling. Its promoter was again studied, and tissue specific expression was found that it was expressing in a leaf specific manner, more in the flower and in the leaf rather than in the tissues like the underground root and the stem. Again, it was having an inverse relationship with various stresses which were found to increase the windowline biosynthesis in the plant. Now we went for a virus-induced gene silencing approach to silence this gene to functionally characterize its role in the windowline accumulation. So we tried to overexpress the gene also, and we tried to silence it also using the Wix approach in the tobacco rattle virus vector. So when we did that, we thought that these changes in the windowline biosynthesis in the plant must be occurring due to the variations in the biosynthetic pathway gene expression. But to our surprise, we found that none of the biosynthetic pathway genes were affected when we tried to silence or overexpress TIL in the plant. So this raised further questions. How could TIL affect windowline biosynthesis when it is not affecting the biosynthetic genes at all? So again, a study was required to localize it in the cell. Where is it residing in the cell? So GFP based fusion tag was made. GFP, you can see here, GFP was fused with TIL at the C terminal of the protein, and it was bombarded into protoplasm to find out where the signal is going. So GFP is a protein which gives a green fluorescence when you observe it under the confocal microscope. So it is just like a taxi driver. It will take your target gene to a place where it would like to go. The driver is your gene, but the signal is GFP. So wherever the signal goes, that means your protein is residing there in the cell. So this particular method proposed that it was a membrane bound protein, TIL was a membrane bound, and also in the internal membranes also. It was found to be located on the cell membrane as well as the tonoplast membrane. So now we had to propose a model which was that whenever this TIL, because it is having a role in the membrane integrity. So this is a normal cell in the center. You can see under the normal circumstances, windowline, which is produced in the cytosol, ultimately gets stored in the vacuole of the idioblast and the latissifer cells. So there is an antiporter here, which with the proton pump, antiporter proton pump, which creates a gradient for this windowline to get inside the vacuole. Now, this cellular membrane integrity on the tonoplast membrane is affected by TIL. So when TIL is expressing normally, certain amount of windowline gets to go inside the vacuole from the cytosol. But when you try to silence this TIL, a more amount of windowline can enter because the membrane integrity is suffering. So the tonoplast membrane suffers integrity loss and allows more of windowline to enter into the vacuole. And the inverse thing happens when you try to overexpress, the tonoplast membrane gets stiffened up, lesser amount of windowline can enter into the vacuole. So now the question was why we are measuring windowline more here in the silencing case and less in this overexpression case. The possible explanation came because windowline is stable in the vacuole. Cytosolic windowline is having less stability than vacuolar windowline. So once windowline gets into the vacuole, it is more stable for a longer time, and we could measure higher amount of windowline in this case. So this is how we could find out the intricacies of sequestration and accumulation of various metabolites in plant cells. So this is one case which we tried to highlight here. It was a challenging study for us. This is another important enzyme. I would like to mention this as a third one, which we tried to work out. Since we all know that the final steps of windowline accumulation occur in the latissifer cells and it gets stored in the vacuoles of those cells, we try to find out what are the relationship between these type of laticifers and windowline accumulation in the plant. So we found out that there was a pectin methyl esterase. Pectin remodeling enzymes like PMEs are known to be involved in latissifer development. There is a functional database for Catranthus roseus genes, which is known as ProFGD. We tried to find out a leaf abundant CRPME isoform that co-express with a few Mendelian biosynthetic genes. It was found to possess all the specific characteristics of a typical plant PNV. It was again characterized using the silencing and overexpression approaches. And comparative analysis of the transcript abundance of the enzyme activity in the three familial genotypes was found. It was higher in Simsuchil than in Hawal than in Lomal. So this study again highlighted the possible role of a enzyme, a cell wall remodeling enzyme in modulating latissifer associated secondary metabolism. Now coming back to the plant, I told that we need to find out genotypes which are accumulating higher than usual level of Mendelian. So what is the usual level of Mendelian? The usual level which we found in our genotypes, in the Indian genotypes that were prevalent at the time of starting the study, was around 0.05% to 0.08%, which is around 50% less than the global benchmark which we had at that time. Specifica piece that produces maximally 0.2% of Mendelian. Hence, a research goal was also established to have an indigenous genotype that could accumulate up to 0.2% or more of Mendelian. For this, the National Gene Bank was searched, which was screened for presence of Mendelian rich genotypes or Mendelian blocks. This strategy was adopted for a long time at CMAP. The Nirmal seeds were treated with EMS, and the plants which were generated in the M1 were chimeric, then they were stabilized. And the N2 segregation population, which we started to screen, that was having around 312 plants at that time. We could find out one particular expression which was high on Mendelian, and that was termed as CMAP 866. But the problem was that there was some need for breeding interventions to increase the harvest. So we tried to work on those aspects for many years, and then ultimately got through selfing homozygous 2 type plants. Then the initial evaluation was done at CMAP Lucknow in 2017-18. And finally, in 18-19, the next cropping seasons, we went for multi-location trials in Bangalore as well as Lucknow. Finally, a new variety could be released in the form of Simsushi in 2019. So these are some pictures of the trials from Lucknow on the top, and Bangalore, you can see the red soil in the middle. These genotypes, if you compare various genotypes, we used Nirmal and Dhawal as the check varieties because Nirmal was the parent variety for all in all the cases, and Dhawal was again a mutant that was developed earlier by Dr. R.N. Kulkarni's group at CMAP Bangalore Center. So you can see the morphology is entirely different. Sim Sushil is having smaller leaves, calyx, and flowers, as well as higher chlorophyll content. So higher chlorophyll content is another question which needs further research to be done, whether it is associated with the Mendelian trait or not. The canopy type as well as the plant height was also very much distinct in all the three genotypes. You can see it is a dwarf phenotype with a very broad canopy in the case of Sushil as compared to the taller Nirmal and Dhaval genotypes. We also did a real-time PCR for checking the gene expression of all the biosynthetic pathway genes in the plant and found that it correlated well with the alkaloid content, specifically Mendelian in the three genotypes. Another interesting thing which came to fore was the elemental composition of the leaves in the three genotypes. Certain elements accumulated to a higher level in the Mendelian rich genotype. For example, if you find that zinc, iron were higher, and boron you can see is lesser in the dwarf genotype because boron is very much needed for plant growth. So it also indicates and provides different directions for research for further action to be done on this plant vis-a-vis the accumulation of important alkaloids. Another picture on the left, you can see the latissifer number or the latissifer count is higher in the middle one, which denotes Sushil. And latissifer number is a function which directly correlates with the Mendelian content in the plant. So we defined the characters, various characters for the three genotypes, mainly the height, stem diameter, internodal, which are very important for characterizing any new genotype which is being developed. And the Mendelian content was found to be much higher consistently at Lucknow and Bangalore. You can see 0.2% on dry weight basis as compared to 0.04 and 0.08 in Dhawal. The total alkaloid content of these genotype was also higher. And again, the total yield was also higher. The harvest was consistently superior to that of the Czech varieties, but it was slightly lesser than the other two at Bangalore. Nevertheless, it produced higher yield for both the alkaloid because the content was higher, It was more consistent in its performance for windolene and total alcohol yield at both the locations as compared to the check varieties. It was found to be equivalent to parental variety label for disease against dieback disease. And all the genotypes performed better at Bengaluru as compared to Lucknow. So that means South India is a better region to cultivate Catharanthus as compared to North Indian regions. Genetic fingerprinting was also carried out to find the presence or absence of specific markers in the genotypes, and a characteristic DNA profile was also generated using 24 decamer primers. I would like to bring here, this is for the benefit of people who are growing Catharanthus or they are planning to, because it's still not very much grown as under North Indian conditions. It is cultivated in about 3,000 hectares in India, mainly only in the states of Tamil Nadu, Karnataka, Andhra Pradesh, Gujarat, MP, and Assam. New areas could be taken up for future cultivation in UP or other states. Through proper dissemination, there is a scope to target around 50% of this cultivation. The cost of semi-synthetic production of anti-cancer alkaloids could be reduced significantly if we have genotypes which are rich in the monomers like windolene. India being a major exporter, around 400 metric tons of leaves of Sadabahar are exported every year on an annual basis based on the data from the Directorate General of Commercial Intelligence and Stats, Government of India. Higher yield of windolene in the Indian export material could fetch a premium price in the market and earn crucial foreign exchange. The USA is a globally the largest user of Catharanthus as a raw material and consumes over 1,000 metric tons of leaves annually. So if you go by a rough estimate, you would need around 40,000 rupees to cultivate the plant in 1 hectare. And if very conservative estimate of 50 rupees per kg, if you are selling the leaves in Lucknow, the yield and Bangalore, the yield of the plant would fetch around prices like 1,20,000 and 1,36,000. So which can give a net profit of 80,000 to 95,000. And this is also has to be considered as a single harvest. Root has not been considered in the calculation. It may be additional, but these things need to be validated also on the field of the farmer. So this variety was released into 2019 and publication, the details are there in the JMAPS issue of 2020, volume 42. Although many genes and enzymes have been worked out, but still do we know everything on this plant? I would say no. Although it would seem strange, I have spent a lot of time working on this plant, maybe around 70 to 90% of my time has gone on this crop, but still I think that that is only a very small drop in the ocean. Still, we fail to understand the plant and the intricacies associated with it. There are several gaps in the elucidation of the biosynthetic pathways for strictosylin-derived alkaloids other than windolene. I also told earlier that more than 3,000 alkaloids come from the same structure. How? It still is an enigma. Besides, there are many regulatory genes that remain to be characterized. Since multiple cell types and compartments are involved in the biosynthesis of TIS, the transporters, their role is very, very crucial. So the next step or the focus on the type of genes required to be studied are the transporters for their role in sequestering these metabolites in various cell compartments in Catharanthus. So we tried to come up with a model in 2022, defining what are the omics resources available in the public domain, as well as what are the transporters which are still needed to be characterized. So we have put a question mark on all the transportation steps from one compartment to other or from one organelle to other. Wherever the transportation mechanism is not clear or the transporters are still to be worked out. Various database resources have come up on this important medicinal plant. More or less, it has become like a model, non-model plant. It is not a true model like Arabidopsis or say tobacco, but among the medicinal plants, it enjoys an exalted status because a lot of literature and genomic resources have been put there in place. As I mentioned earlier, there is a very good database known as the CRO-FGD database by a Chinese group. They have put together all the information available on this plant in various resources and tried to bring up a co-expression network exploring multi-dimensional gene expression. Then there was CathaSeq, which was also a metabolic pathway database from the RNA-seq data on the plant. From CMAP also, another colleague of mine, Dr. Bhaskar Shukla, he has tried to put generate a MAPS LNC database, which is the repositories of long non-coding RNAs on the plant and other medicinal plants also. So there is a list which one, if one is interested, they can go through this publication. They will find all these things there. And I have also tried to list the challenges which need to be addressed in future research on this plant. There is a need still to create genotypic diversity on the plant through various approaches, mainly plant breeding. I still feel classical plant breeding is much more helpful than going through modern approaches, but this statement I would like to say both have to go hand in hand. Neither alone would be able to go a far way. We have to work together using the classical approaches as well as the modern approaches. But in isolation, either won't work well. The elucidation of many pathway genes for other TIS. We only focus our studies on the bis indoles, on the monomers, but there are more alkaloids. 130 alkaloids still need to be studied for their biosynthetic pathway. Analysis of the metabolic flux channeling into various branches originating from streptocittin, the central intermediate, the chameleon, which I talked about, is still very much required. The complete arsenal of alkaloids is yet to be completely elucidated. Novel alkaloids with unique therapeutic properties present in the plant might still be waiting to be isolated and characterized. Since microRNAs are crucial regulators of gene expression, further investigations are needed to elucidate the regulatory roles of miRNAs and even the long non-coding RNAs in the plant. Novel methods are still required to circumvent the recalcitrance of Catranthus roseus to establish transformation methods. There are several attempts at transforming this plant. Some successful attempts are also being made. They're also being made at CMAP, Dr. Mathur's group. Others are also there. But again, methods need to be more robust, and there are still novel methods required for circumventing this type of recalcitrance of the plant to transformations. So one very good example came from the group of Mortimer et al., who exploited the replication system of Catranthus infecting Gemini virus towards development of an autonomously replicating viral expression system tailored for Catranthus. The influence of various seasonal and ontogenic parameters on the pattern of TTIA accumulation has been well established by now by our group, and agro advisory could be suggested to the commercial growers of the crop for appropriate harvesting time for specific TIAs. Although cellular differentiation, elicitation, and biotic abiotic stress factors are known to influence TIA biosynthetic steps, a holistic picture still eludes us. The exact mode of action of each type of stress factor needs to be worked out for better understanding of TIA metabolism. Comparative analysis of Catranthus must also be carried out with other species in the genus Catranthus so as to identify the similarities and differences. Why is Vindolin produced only in Catranthus roseus and not the other species of Catranthus? Those type of questions, if answered, we can use these species and get very good information. Catranthus has high potential for phytoremediation of toxic metals in sewage sludge, acts as a good host for phytoplasmas infections, and leaf extract offers a good choice for synthesizing zinc oxide nanoparticles for miscellaneous applications such as antimicrobial agents. So these are some of the other added advantages of the plant apart from its therapeutic value, which also needs to be exploited. Since we focus too much on the bis indoles and the monomers, I thought that we are not doing justice to the other alkaloids which are also present in the plant and which are also very, very important. So I try to list some of the other alkaloids which are also having some therapeutic activity, but they might not be as prominent as the others which we talked about recently. Yohimbine is an antiviral compound present in the plant. It has effect on the herpes simplex virus with a cytopathogenic effect at 0.8 micrograms per ml. It also shows antifungal effect against the opportunistic pathogens Candida albicans. It has been prescribed as a treatment for erectile dysfunction also, although its reported clinical benefits were modest and have largely been superseded by the PD5 phosphodiesterase type 5 inhibitor class. Cathorasumine is a potential inhibition against Plasmodium falciparum, which is a positive agent for malaria. Azmalicin is an antihypertensive, acts as an alpha androgenic receptor antagonist. Serpentine is a root alkaloid from the plant, antihypertensive, acts as a potential anti-neuroinflammatory agent since it inhibited the receptor of cyclooxygenase 2. Reserpine is again a hypertensive, again a root alkaloid, also produced in the sister species Rauwolfia serpentina. Vincristine and vinblastine are known for their anti-cancer activity, but interestingly, they also have anti-parasitic effects against Trypanosoma cruzi, and that causes trypanosomiasis in humans. Vindicine is again a semi-synthetic derivative from vinblastine, which provokes the arrest of cells in metaphase, just like the other bacillus, vincristine and vinblastine. Vinfluinin, it is a first fluorinated microtubule inhibitor synthesized from vinorelbine, but it has lesser neurotoxicity. So all these alkaloids which are being generated semi-synthetically, the target was to reduce the toxicity of the parent compounds like vincristine and vinblastine because they had serious side effects. So everyone wanted to reduce their toxicity so that they are having higher therapeutic window, higher therapeutic value, and lesser of toxicity. Vinolene, again, the monomer has hypoglycemic activity of its own, as I told earlier also. So here I would like to thank all the research scholars who have been associated with me over my journey. Dr. Priya Nair, Dr. Manisha Mall. Manisha worked mainly on Cathanthus for a long time. Mridula Singh. Priya worked on Artemisia annua, Mridula on Papaver somniferum. Devas Pratap Singh and Amit Kaushik, they work on Acaranthus aspera, various other aspects. Pooja Singh was instrumental in the variety Sim Sushil. Seema Yadav has been working on micronutrients on the plant, specifically the role of various micronutrients in defining the alkaloid profile in the plant. Mridula Sahu is working on intestinal bowel syndrome disease. Pratibha is working on Tylophora, another plant. Kajal Kumari has recently joined the Catranthus group. So you can also see there is a membrane and everyone has to cross the membrane. Some of them have crossed successfully, others are in between, and some have started their journey. So I would like to thank Director CSIR CMAP for all the support which has been provided to me for my research, my team at CMAP, my collaborators, and most importantly, the funding which I have received from CSIR, Science and Engineering Research Board, PST, DBT, ICMR, and I'm open for any questions I would like to answer if I can solve the queries if I have raised among the audience. Thank you very much.
Thank you. Thank you, Dr. Ashutosh ji, for a very, very fascinating lecture, very informative lecture. I would request our founder president, Professor Khanuja, for his remarks. Sir, sound please. Sir, unmute yourself.
Thank you, Dr. Singh. Thank you, Ashutosh, for bringing so much of depth of science to the knowledge of all the audience today we have on this topic. Sadabahar, although is known to everyone, but the depth of metabolism and the molecules that it carries, today you could bring that up to everyone. Before I come to any discussion, since your presentation has been highly scientific and would be very useful to the students and researchers, I'd like to put out some of the points that common man would like to know about it, which came out of your presentation. So first was drugs are poisons. Herbals, usually people start using on hearing that this is very useful, and they even sometimes start eating like food. And you did point out that how poisonous it can be and how toxic it can be when you do not have the proper knowledge of the content of the material that you are eating, dosage and toxicity relationship there. So therefore, one has to be really careful for utilizing the plants, not just by hearsay, but by understanding what the plant contains. So while Sadabahar is useful for many, many lifestyle diseases and going up to cancer, but still the use has to be on the basis of what has been recommended medically, and also even following traditional knowledge, what you told, again, dosage and the process of preparing that drug becomes very important, and that is well elaborated in our ancient literature. Some of those you also gave as the hints to the people. Second thing that comes out for understanding of every one of us is that Catharanthus shows the metabolic pathway circuitry. The circuits that we see in a computer like ICs, and the metabolic spread in the pathway branching and pathway merging, making it like a real internet of the metabolites, is a very good example that would tell us that how biological circuits can be produced and utilized in future, particularly for AI purposes, and also utilizing AI for medical purposes. So that is going to be an area where Catharanthus can be a very good candidate for researchers. Third thing I felt what you told every one of us is that plants have feelings. Plants feel pain. And the example that you gave is from monomers like catharanthine and vindoline going into the dimers when there is a damage to the tissue. You showed very good picture of a sheep grazing on plants. And when that type of damage happens, they shift their metabolism. So that's what the feeling is. The chemistry of plants is the feelings that gets expressed by diversion in the pathways. And after damaging, if we are going to harvest the plant materials, then they may be chemically very different than if we know the harvesting technique. So therefore, even for harvesting, we have to value the feelings of the plants and the pain of the plants. So it depends on what really we want to harvest. And therefore, such things can also be utilized as switch to utilize the plant materials in the process itself, right from harvesting stage. Our last theme was fasal se safal. So that fasal is the harvest, which if made very systematically can give you the right quality, which you mentioned, can fetch us the good export value also commercially. And that is where the economics that you had shown in your table would be dependent on. So although in a analytical method, we might have got some yields or figures that this is the percentage of a metabolite, but then how did we harvest and what material changes in the metabolism have taken place will determine the quality. And that's good for the industry to learn from your presentation. And one more message which comes is that, like all plants, particularly the plants which are highly medicinally important for phytoceuticals, or the molecules that are going to be used for multifarious drug values, age, form, tissue, organ, and its condition. That means whether diseased or disease induced or some damage happened, all this becomes very, very important to think of an industry where we even use plants as bioreactors. So therefore, those things, if we really take care of, we will also have to consider the last few slides that you showed, the localization and transport of these molecules across the membranes. So those things will become very important. And I feel this is going to be the basis of future synthetic biology research with all that knowledge that you talked about in form of an AI or AI tools, and making into a field of synthetic biology which really works at industrial level. Now, with these, some of the messages that your lecture has given, I would like to pose you two questions. First question is, your topic is traditional to modern medicine. Now, considering the knowledge that we have, traditional wisdom-based knowledge, which tells this plant is useful for these diseases or ailments. and the molecules, and then the drug validation of those molecules, which has happened in modern biology. Where do you see the gaps between traditional knowledge and modern molecular-based medicine in case of catharensis?
So this is a very good question and a difficult one also for me to answer. But the thing is, uh, traditional is holistic. You take the whole thing, and modern medicine is a reductionist approach. So I think always we cannot move from holistic to reductionist. Sometimes, as in the case you also told that the traditional systems describe the mode of purification for reducing the toxicity. So many a times you cannot do away with the toxicity. Toxicity has to be there. Only thing is you can reduce it. So there are Shodhan methods described in Ayurveda for treatment where these neurotoxicity of these bacillus could be reduced, but it cannot be done away totally. So the bad thing also comes with the good. So we have to have a balance everywhere. The middle path is the best path as told by Lord Buddha. So only thing is the modern medicine identifies the therapeutic value only when you pinpoint that a certain metabolite or a molecule is able to give this activity, which is not the case always in traditional systems. Traditional systems, you use extracts or the plant parts as such. You just know that by experience or hearsay that this is not toxic. And so the tribals are using it for a long time. Many a times that people will try to find out a specific species which is identified only by the tribals and nobody else can identify also. So they have used it for ages and found that it is working. But again, modern science and biotechnology gives a caution that the place where we are going the plant, how we are getting those metabolites when we are harvesting, the environment plays a very major role. So I think both the approaches should go hand in hand. Traditional medicine will provide you the cues, but it is the responsibility of modern medicine to test them and bring out the facts before the people. Then the people will decide for themselves what is good for them. But again, people should not start consuming the plants on their own just because somebody else has told them to do so. That is my opinion. I don't know whether I have answered because it is difficult to answer a teacher when he poses a question.
No, it's not like that. You very rightly put traditional versus modern medicine systems. But my question aims at, there are potential drug values which traditional system has mentioned and potential drug values which modern system has mentioned. Are there gaps or they are similar? In utility only. I'm not talking of, how much or how to use.
So there are gaps. Gaps in the sense that traditional system will tell you that you can use the leaves of Sadabahar. But how many leaves are to be consumed on a daily basis and which leaves are to be used, we don't know about it. So a person will start growing Sadabahar in a pot and then every day he will or she will be using certain number of leaves, which might be deleterious. So that gap has to be filled by the traditional system also. And in the modern system, still there are so many gaps. We are only focusing our activities on 3 or 4 known alkaloids. There are more than 130 alkaloids. Each one of them needs to be separated, its structures worked out, and then their activity tested. So it's a long term. Both have their gaps, but they are different, which needs to be filled through research only.
Okay, so I'm seeing Dr. Mani also sitting here and listening. And Dr. Mani, if you can put
Yes.
on your
Sir, good afternoon, sir.
good afternoon. Now, let me introduce the audience to Mani. He is the Ayurvedic expert and has been now working with the CMAP and therefore well exposed to what modern biology says. So, Mani, do you have any comments to offer now?
Uh, Really, sir, I agree with Dr. Shukla. The traditional system use the medicinal plant before purification. Purification means शोधन. तो आयुर्वेद में भी सर जो भी ड्रग्स हमारे हैं वो शोधन आफ्टर प्यूरिफिकेशन के बाद ही कंज्यूम करने की बात कही गई। सेकंड ये है सर कि उसे जो मेडिसिनल प्रॉपर्टी बताई गई है आयुर्वेदा में, सीमैप बेसिकली उस तरह के काम कर रहे हैं जो होलिस्टिक मैनर में है। तो स्पेसिफिक अगर किसी कंडीशन में तो उसको साइंटिफिक हम लोग वैलिडेट करने की कोशिश कर रहे हैं और उस बेस पे चूंकि कुछ ड्रग सीमैप जो है वो डेवलप कर पाई है सर।
Okay, I also see Dr. Ravinder Rana sitting here, listening to this talk. And Dr. Rana is a well-known expert of medicinal plants and also a entrepreneur in that field. Perhaps he might have a question from herbal point of view to Dr. Ashutosh Shukla.
Uh, Dr. Shukla, it was mind-boggling lecture because many of the things which you said were new to me. And this naturally leads me to to the question which Dr. Knuja has already raised, that at least when it comes to Sadabahar, this should be off the table for the traditional medicine because the kind of toxicity and the varying range of alkaloids it has to be taken with a caution and a precaution. And generally, when the traditional medicine, especially on the basis of hearsay, when people use it, they may end up with more trouble. Uh, there's a common practice, especially by the diabetics, putting the flowers of this plant in the water and for keeping it overnight and drinking the water next day. Can you please tell me how far is that correct? Is it just a psychological effect they are getting, or is really some leachates who come out in that water, and they have some effect, and that which has some effect on the sugar content?
Thank you, Dr. Raina. You have raised a very pertinent question, and I don't have the answer for that per se because whenever you are using any tissue, keeping it overnight and using it, to give a scientific answer, that extract has to be chemically defined. It needs to be validated through LCMS or metabolites which are present therein need to be identified. Then only a scientific answer can be given. But again, as I told, in the traditional systems, always modern science cannot immediately interfere. People have been using things for a long time and successfully. So that means there are benefits in traditional system also. Only caution is that don't go by hearsay. Only if something is mentioned in the classical texts of Ayurveda, say, then you can definitely rely on them. In Dravgun Vigyan, which I already mentioned in my presentation, Acharya P.V. Sharma has described this for treatment of diabetes. But that plant has to be grown in a certain location. It has to be cultivated in a certain climate. You cannot use any plant which is growing anywhere because the metabolic profile changes. As I told in our case also, Sim Sushil is performing better at Bengaluru than in Lucknow for the same metabolite. That means plant is producing windolin, but the content might be very much different. So it's not a very ready-made or easy solution to answer this question. A lot of effort is needed to chemically define a window for the metabolites and their activity or toxicity per se to define a safe value for human consumption. And this is not the problem with Catranthus. It is a problem with every medicinal plant which is being used by.
Almost, yes.
So you have to be cautious everywhere. So I'm sorry, I'm not able to answer that further, but a lot of research effort is still required. I also told that it's only a drop in the ocean, which I know.
But the way you have presented the whole lecture was very interesting. It's an eye opener in many ways
Thank you.
because the kind of diversity and range, and definitely I also agree with you that this should not be the problem with only with catharanthus, but should be problem with most of the plants that we use, especially those of the plants that we use over the counter, and that we have to be very careful about it.
Another
And there are
example is like opium poppy. Opium poppy is also having good and bad effects. Everyone knows morphine is produced there, but it is also abused as a narcotic. But again, Ayurveda mentioned its use as I think Afim is used described in classical texts of, I think Dr. Mani can elaborate on that, but after Shodhan only. So that Shodhan brings a lot of changes in the property, and those biochemical reactions have to be studied. Nobody has attempted to study the process of Shodhan or the process of formation of these Ayurvedic things because they are not depending on the present metabolites in the plant. They give rise to newer compounds and newer biochemicals after those processes occur. So there is a need to have a Ayurvedic biology program, which was earlier also there, but again, these type of things also need to be looked into in a deeper manner. That is my say on this aspect.
I mean, that leads us to a much larger question, and well, generally we also we so medicament vis-a-vis the modern medicament. We should be okay with using the traditional medicament for preventive purposes, the prophylactic approach. And when it comes to the therapeutical approach, the curing part, then we should use these with a bit of more caution. More caution is required in using them, because as you have brought out nicely in case of Tadabar, the situation may be present in many other plants also. So we should be, as far as the research approach is concerned, the prophylactic approach also needs to be validated. And that is probably a bigger strength this traditional medicine has than the therapeutical. With the therapeutical refined chemical and alkaloid, that will give us much more scientific to address a situation, to address a disease situation.
I agree, sir. Actually, the problem is even in Ayurveda, say if you go by the traditional system, it has been, we did an earlier project under the leadership of Hanuja sir. It was a science initiative in Ayurveda project when Professor Valiathan was leading the task force. That time, we try to work it out from the plant perspective. The plants are used for their metabolites present therein. But Ayurveda has also described specific seasons for the collection, specific locations for the growth. So that specific season and collection pattern or the things described in the traditional text at least ensure that a proper combination of the metabolites are present in the plant. But nowadays, what is happening, the climate has also changed. So even if you are extracting the plant in the defined climate, it might be raining in the month of, say, June heavily, or in May also. So the metabolic profile changes immediately. The plant takes a fraction of a second to change and decline because it is sessile. It cannot move. It has to stay at its place. So these things are not so simple. We debate and we discuss so many things, but still we know very few things. Science still has a long way.
Yeah. That's that. Especially we should be more careful with the exotics, which we have domesticated in our country, because they come from a different climate and geography, and they may be doing well as far as the growth is concerned, but their omics profile would be totally altered, would get totally altered. So we have to be careful with those.
I mean, there is no magic band which we can answer these questions, but
Yeah.
if we start thinking about them, and people should be aware that just eating the leaves won't get you rid of diabetes. You need to have a proper validated regimen.
Yeah. Yeah. Very impressive lecture. Congratulations for a very good talk.
I think Dr. A.K. Singh would like to conclude.
डॉक्टर शुक्ला, आपका लेक्चर बहुत ही जानकारीपूर्ण रहा। एक जो शुरुआत में आपने बताई थी जमाइकन टी के बारे में,
जी सर।
इज इट स्टिल बीइंग यूज्ड बाय दोज पीपल, और वो क्या इस्तेमाल करते हैं, फ्लावर और लीव्स?
सर वो लोग लीफ यूज कर रहे हैं।
अच्छा।
लेकिन अब उनका ये भी है कि वो कहीं पे उन्होंने डॉक्यूमेंट उस तरह से नहीं किया गया है।
ओके।
कुछ लोग फ्लावर भी यूज करते पाए गए हैं, और कई लोग डायबिटीज में कोई कहेगा फ्लावर अच्छा है, कोई कहेगा लीफ अच्छी है। लेकिन ये जरूर है कि सर्टेन एल्कलॉइड्स प्रेजेंट इन द लीफ, लाइक विंडोलिन का मैंने एग्जांपल दिया, हम लोग उसको डॉक्टर मणि की सहायता से वैलिडेट करने की कोशिश भी कर रहे हैं। तो उसमें है हाइपोग्लाइसेमिक प्रॉपर्टीज।
ओके।
कंपेरेबल है कि नहीं मेटफॉर्मिन से या ड्रग्स हैं, वो चीजें हमें देखनी।
क्योंकि जब ये पौधा हर जगह ऑलमोस्ट एवरीवेयर इट इज फाउंड ग्रोइंग, तो किसान लोग भी कहते हैं भाई इसका फ्लावर पानी में, जो भी संदर्भ आया था, डॉक्टर रैना ने भी जिक्र किया कि पानी में रात भर भिगो दिया और उसको सवेरे उसका पानी ले रहे हैं।
सर इसका आंसर देने के लिए आपको एक एक्सपेरिमेंट सेटअप करना पड़ेगा कि
होगा
ये उसको पानी में मत भिगोइए, एक को भिगोइए, तो उसके बाद आप कंपेयर करेंगे तो सारे आंसर्स निकलेंगे। लेकिन ये अच्छा आपने पॉइंट दिया, इसको हम लोग सोचेंगे, आगे विचार करके इस पे काम
जी
करेंगे।
जी जी। आपका लेक्चर बहुत ही जैसा मैंने कहा कि जानकारी पूर्ण रहा, एंड वी आर ग्रेटफुल टू यू फॉर स्पेयरिंग योर वैल्यूबल टाइम, एंड वी हैव I mean, initially we found some difficulty to find your dates because you were busy in your research and other engagements also.
I'm sorry for that, sir.
No, no, thank you very much for sparing your valuable time and giving this very informative and fascinating lecture on traditional medicine से जो आपकी यात्रा इसमें शुरू आपने बताई शुरुआत की, और मॉडर्न मेडिसिन तक, और भी आगे क्या होना है, और ये भी बताया कि किस प्रकार से इसका कल्टीवेशन करीब 3,000 हेक्टेयर में हो रहा है साउथ इंडियन स्टेट्स में. सो दिस इज ए गुड साइन एंड होपफुली दिस विल इंक्रीज. दिस इज बीइंग एक्सपोर्टेड आल्सो, बाहर भी जा रहा है करीब 400 मीट्रिक टन्स की क्वांटिटी में. बहुत बहुत धन्यवाद आपको. All the participants who have joined today, listeners who have joined our Facebook, all the students and researchers, farmers, सभी के हम आभारी हैं इस मंच पर आकर के और हमारी हौसला अफजाई किया है. फ्लोरा फौना साइंस फाउंडेशन जो ये मंथली वेबिनार आयोजित करता है, वास्तव में इसकी सफलता के पीछे आप सबका पार्टिसिपेशन है. बड़ी से बड़ी संख्या में आप पार्टिसिपेट करिए, और उसके बाद भी ये जो लेक्चर्स हैं ये फ्लोरा फौना साइंस फाउंडेशन की वेबसाइट पर उपलब्ध है, बल्कि इसमें फेसबुक पर भी उपलब्ध है. आप इनको सुनिए और लोगों को भी बताइए. वी आर थैंकफुल टू यू. वी आर थैंकफुल टू प्रोफेसर रैना, Dr. Mani for their inputs, and all the scientists who are present here on the panel. It will be difficult for me to take name of everybody because it will take a lot of time. Professor Khanuja, who has been guiding spirit for holding of these webinars and selecting the speakers one after the other. Thank you, sir. Dr. Manika, who has conducted this Zoom meeting, we are thankful to her also. And with this, we will be closing this webinar part. And next one, we shall be meeting with some new expert, एक नए विशेषज्ञ के साथ, एक नए विषय के साथ. तब तक आप सभी का बहुत बहुत धन्यवाद. नमस्कार. Stop it.